Hello, Harshraj.
Yes, sir.
Yeah, so you sent me a question, I just checked.
Yeah, the answer I checked cross-referenced, and it seems to be the suggested answer. It's showing B, which in my list it's appearing as enzyme substrate
No,
complex.
actually, I also, after reading the question, I also felt the answer should be A, but I don't know why is it giving B as the correct option.
There's some
Um,
administration error. That's
yes, what?
I was just thinking, ki, how does this like influence like our grades if we get it wrong in our assignments? Is there any way?
I don't think that because this is just an assignment. You have to give an exam to be
Yeah,
graded.
I know, but there's 10 out of 25, 25%, no? Like that will be done, but still that score will be added right around
Okay, so
7.
but okay. Anyways, I think one point, but I feel holoenzyme should be the correct option. Because apoenzyme and coenzyme gives you a holoenzyme only. Enzyme substrate complex, because that is on a very general basis. It is telling enzyme substrate complex. You get an enzyme substrate complex anytime, but that does not mean that the answer, when you're specifically it's given apoenzyme and coenzyme, I feel it should be a holoenzyme only. Okay, so I feel A is the correct option. Maybe they gave here something incorrect. Probably, I don't think. Maybe I will ask the course coordinator to just recheck it once again. Okay.
All right, all right.
Yeah.
Thank
So
you.
then maybe no points will be deducted for this one.
I'll try to keep my eye out if I can see more errors. I was just going through a few of the assignments. I'll just see if I can flag more of them.
Means I did not get you.
Like I'll just see if there are more errors. Like I think 95% like it's all right, but like one two places more I thought ki this could also have been the answer, or this could also have been an answer that way.
Okay. Okay. Never mind. I will just cross check with the course coordinator.
Yeah, yeah, yeah.
So I think we can start with today's lecture. If anyone else is coming in, they can join later. Okay, so hello and welcome everyone to today's live class session of week 10 on the course of Fundamentals of Protein Chemistry, which is being taken by Professor Shagata Dasgupta of IIT Kharagpur. So I will be discussing with you the lectures in brief, and then solving a few questions that will help you in solving your assignments and the future exams. Okay, so I'm sharing my slide with you all. So is my screen visible?
Yes, sir.
Okay, good. Okay. So today we would be mostly discussing about protein macromolecule interaction. That is, there are so many. We have studied about protein ligands, means interaction between protein and something else. We had studied about enzyme substrate interactions and all. So today it's about a protein is there, and then there are macromolecules. These macromolecules can be your carbohydrates, can be your lipids, can be other small molecules as well. But there are small molecules. So these are big molecules. So in the big molecules, you have the carbohydrates, lipids. They can be other proteins as well as they can be nucleic acids. So in today's class, we would be discussing mostly about protein carbohydrates and protein nucleic acid interaction. So if we talk about protein carbohydrate interactions, so these are basically your intermolecule and intramolecular interactions that occurs between a protein and a carbohydrate molecule, and these interactions form the basis of specific recognition of carbohydrates, mostly by specific kinds of carbohydrates, like you have the lectins. Now these carbohydrates are important biopolymers and have a variety of functions, like in recognition element, in cell signaling pathways, in different processing of downstream signals and all. And this is recognized by other biomolecules, like you have the proteins. So these proteins which binds to carbohydrate structures are known as the lectins. So compared to the study of different protein-protein or the protein-DNA interactions, it has been uh, seen that scientists are in today's world, or over the past few decades, are mostly interested into this protein carbohydrate binding, and has been the very hot topic. Now you would be seeing that many of these protein carbohydrate interactions are found at the cell surface. So this interaction is occurring at the surface of the cell as a part of the membrane glycoprotein or the glycolipid, and these play different interactions in your cell adhesion or different cellular recognition events. And these intramolecular carbohydrate protein interactions between your glycan and the polypeptide moieties occurring glycoproteins, or you can say glycosylated proteins. Now this is your classification of different protein carbohydrate interactions, or there are two types of protein carbohydrate binding that is important. So the first one being your lectins, and the other one is your antibody. So what are lectins? So these are the kind of proteins that bind to the carbohydrate via some recognition domains or recognition motifs, which you call as the carbohydrate recognition motifs. And we could and there are different kind of lectins. So you have the C type, P type, and the I type. So in the C type of lectins, you have the calcium ion that is required for the activation of the binding process, and this calcium binds to the protein and the carbohydrate via the non-covalent bonds. And your, for example, you have the mannose binding protein that contains the C type of um, carbohydrate recognition domains. The other important type is your P type, and two types of mannose 6 phosphate can recognize your phosphorylated saccharides. One is the cation dependent and the other one does not require a cation to activate the binding process. You have also an I type of lectins, which is named from the immunoglobulin-like domains. Like you have the adhesine, which is a type of I type lectin that binds specifically to the sialic acids. So this is one category of classifying the protein carbohydrate binding, and the other one is your antibody. So most antibodies have similar kinds of structures except the variable region which is called the antigen binding site, and this region is mostly a combination of different amino acids that is important for capturing a particular antigen. And when the antigen is a kind of a carbohydrate, like you have a polysaccharide, then the binding could be termed as your protein carbohydrate interaction. Now there are different functions of protein carbohydrate interactions, like they play different roles in cell adhesion, then signal transduction, host pathogen recognition, stabilization of the protein structure, and inflammation. We have different methods for studying these protein carbohydrate interactions, one of which we have the X-ray crystallography, and to understand the detailed information about the interface of this protein and the carbohydrate, and to get detailed information about the interaction between these two macromolecules. The next one is a very interesting study, that is the NMR, or the nuclear magnetic resonance study, where you use, via the titration, is the nosy kind of experiments like nuclear overhauser effect spectroscopy. So that is the nosy. That is a kind of an experiment which we perform in NMR studies. You have others like CIDNP experiments, where you study the specificity and the affinity of the binding via the titration studies. You get an information about the association constants and different thermodynamic parameters like the equilibrium constants and all about these particular interactions. Apart from these two interesting studies, you have other studies like molecular modeling, then fluorescence spectrometry, and you have the dual polarization interferometry. Now let us get accustomed to a few of an interesting classification of carbohydrates. So this is the first one, being your glycoconjugates. So what are glycoconjugates? These are basically covalently linked with other chemical species, like you have, in this case, you have proteins, then there can be peptides or other kinds of compounds. And in the category of glycoconjugates, you have glycoproteins, then glycopeptides, peptidoglycans, and these glycoconjugates are basically involved in cell-cell interactions, recognition of different motifs or domains. They perform several important roles in cell matrix interactions and in the detoxification processes. So this is your PLP bilayer, where you have the phospholipid. This portion is a phospholipid bilayer, and these are the different membrane proteins, can be integral proteins or the peripheral proteins that you have over here. So these are attached to different glycoproteins, and this one is your glycoprotein, where you have the protein over here, and this is the protein, and this is the carbohydrate. Also you have the lipid over here, and this is the carbohydrate, so you can call this as the glycolipid. And as you can see that these processes are mostly occurring on the cell surface, or on the outer portion of the cell, because this is portion is your cytoplasm. There are different cellular machineries that are functioning over there, and there are different downstream signals that are passed on when there is a recognition over here, or when there is a capture, and there is a signal that is passed inside the cell. So this is these interactions are mostly occur, say you call it adhesion or recognition. Everything occurs on the outside of the cell, which we can observe for glycoproteins, glycolipids, or other carbohydrate interactions, macromolecule interactions. Now you have the glycoprotein. So the carbohydrate is the glycan portion, that is the covalently bonded to the polypeptide side chain of your proteins. Then you have the sugars in glycoproteins that are attached to the amide nitrogen of asparagine, which we call as the N linkage, and the oxygen atom of serine or threonine, and we term it as the O linkage. So one glycoprotein hormone that is mostly present in the blood serum that enables the production of your red blood cells is your erythropoietin. We are very accustomed to this particular glycoprotein hormone. Okay, so most blood groups like antigens are glycoproteins, and the specificity is mostly determined by the oligosaccharide that is present on the surface of your red blood cells. So what are glycoproteins? So these are oxygen-linked glycoproteins, and these are N-linked or amide-linked glycoproteins. So the amide linkage occurs via the asparagine residue, and your oxygen-linked glycoproteins can be because of the serine or a threonine. So if we speak of the structure of glycoproteins, so these are basically the proteins that contains covalently attached sugar residues, where the hydrophobic and polar characteristics of sugars may dramatically change the chemical characteristics of the protein to which they are attached. Now, the addition of sugar is often required for the glycoprotein to function properly and reach its ultimate destination in a cell or an organism. Now, this is very important where we speak of the functions of glycoproteins. So, the first one is that glycoproteins provide structure. So, like you have the collagens, it provides a rigid structure to your cell or to different say proteins and all. And there are some glycoproteins that are involved in immunity. Example, you have the immunoglobulins IgG, IgA, IgM. You have the different family Ig superfamilies. Now you have the mucins that are secreted into the mucus of respiratory and the digestive tracts, where these specific mucins can retain water, allowing the mucus to serve as an effective lubricant. And the other type is to determine the blood group type. So specific glycoproteins present on the surface of the red blood cells helps you to get an idea about what blood group type you contain. This is very important where we come to where we speak of lectins. So these are a type of proteins that bind specifically to your carbohydrates, and they might bind to a soluble carbohydrate or to a carbohydrate that belongs to a glycolipid or a glycoprotein family. Now, these lectins usually contains more than one binding site for different carbohydrate units, and these typically agglutinate your red blood cells. Therefore, you can term these or call these lectins as hemagglutinin. So there are different plant lectins, like you have the canaveline A, you have the ricin, you have the aberrine, you have Wheatstone agglutinin, then you have soybean lectins, and you have the peanut lectins. So there are different classes of lectins that I was speaking about. So the most two important being your C-type and the P-type of lectins. So the C-type lectins requires a calcium ion for binding to a particular carbohydrate, and if you can remember, it's a non-covalent type of bonding. And these calcium ion will link the sugar residue to your lectin macromolecule or the lectin protein. Now the other type is your P-type lectins that contains a phosphate group. Now, very important functions of lectins includes your the facilitation of your cell-cell adhesion or communication. That is, it enables your cell-cell contact and It passes on different signals and feedback inhibitions to the inside of the cell. Now, you have the beta carbohydrates on the surface of one cell that binds to the binding site of lectins on another cell, and these can be sometimes important, as well as sometimes this is a message to the cell that no, this is an foreign material and this needs to be damaged. And in this manner or the other, you have your nectins and then cadherines on the cell surface that actually passes the messages to the inside of the cell, and from there on it can actually regulate your B cell production or the T cell activations, okay. And this can lead to the formation of say NK cells, which is going to damage your foreign materials and all. So this is on one way it enables your adhesion and contact and as well as communication. So the binding results from numerous weak interactions comes together to form a strong attraction. Also, lectin usually contains two or more binding sites for a carbohydrate unit. And in addition, the carbohydrate binding specificity of a certain lectin is determined by the amino acid residues that binds to the carbohydrate. So this is a cell surface oligosaccharides. This you can term as lectins. So this is a cell one where you have the cell surface oligosaccharides, and this is your cell two. So if you can see over here that this is your fit model, induced fit model. So you can also call it as the lock and key model. So only those two, you can say the lectin and the carbohydrate is going to come together where there is a fit of structure. So this is what you observe in cell 1 and cell 2, and this is the binding of the lectin to your oligosaccharide. So in this way the two cells comes together and communicate. The other type is your, not the other type, but the C-type lectins is your, also you can call them as collectins. It's a part of your innate immune system. Their function is to bind to oligosaccharides on the surface of microorganisms. And the binding of collectins to microorganisms may trigger elimination of different microorganisms, foreign particles by aggregation, then complement activation, opsonization, or the activation of phagocytosis. In this way, you can you trigger your activation of B cell and T cell production and NK cell production, and in one way it can grab your foreign materials or microorganisms via your phagocytosis, opsonization, etc. Then you have the selectins, which is also a C-type lectins, which play a role in the immune system by the recruitment of your WBCs into an inflamed tissue. So these, what does these WBCs do is that they do not interact directly with the blood vessel endothelium. Okay, so these inflammation causes the expression of selectins that on the surface of the blood vessel wall. And then these selective carbohydrate units on the surface of the WBCs present in the flowing blood can interact with your selectin, and this interaction, or when they come close to one another, it results in the rolling of the WBCs on the endothelial cell surface, followed by a stable adhesion and migration of the WBCs into your inflamed tissue. So you can see over here you have a selectin. Here is the WBC that um, presents a carbohydrate on the cell surface, and this is an inflamed tissue with the migration of the WBCs into the inflamed tissue. So it comes in contact. The first step is the WBCs presents its carbohydrate, and then you have the selectins on the surface of the endothelial cells. They come in contact and then they go inside and undergoes the repair mechanism. So the functions of lectins, lectins are stored in seeds, which function as your storage proteins. They function in your defense machinery. When an invading microbe or insect disrupts the plant's cellular contents, the stored lectin may agglutinate or sicken the pathogen or a predator. Now, your ricin is a toxic lectin that is present in castor seeds, which inhibits protein synthesis at you say ribosomes. Then you have the lectins that binds the either glucose, mannose, or different you can say uh, sugars, like you have the galactose. Now they are involved in the attachment of nitrogen fixing bacteria to the legumes and in the protection against different pathogens. So that was all about our okay. So that was all about our protein carbohydrate interaction. Now we will be speaking about the protein nucleic acid interaction. So this protein interaction, nucleic acid interaction can be a protein DNA or a protein RNA interaction. And any of this protein nucleic acid interaction is basically termed as electrophoretic mobility shift assay, or in short we can tell them as EMSA. So it's a powerful technique for detecting a specific binding of a nucleotide to a protein, and we are studying the complex. It can be the binding affinity or the specificity, and it is done by this technique known as EMSA. So, what over the past few years, IMSA has been the go-to asset to investigate the qualitative interaction between your DNA and RNA with your proteins, and it's a very rapid and sensitive method to detect the protein complex with a particular nucleic acid. So, what is basically this technique, or what is basically this term IMSA? So it is known as your gel shift assay, or you can tell as the gel mobility shift assay, band shift assay, or a gel retardation assay. So we, in this particular technique, it is to detect whether the protein that we are having in hand is binding with your DNA or RNA or none of the above. So we know that there are many cellular processes where the protein needs to interact with the DNA, like you have the transcription and the translation. You have different transcription factors, and as we know that the transcription factors are nothing other than proteins that binds to your DNA sequence. So it can be a promoter sequence, enhancer sequence, and the silencer sequence. It can be any of them, and it regulates the transcription as well as the translation of a particular a set of genes. Now, it's originally utilized broadly in the investigation of sequence-specific DNA binding proteins, such as your transcription factors, and it has been additionally developed to explore the DNA protein interactions and RNA protein interactions. And as I mentioned, it's a very sensitive method using radio labeled. And so these DNA or RNA is basically radio labeled, and it is made to interact with your protein, and in that way you can study the protein DNA complex formation via the autoradiogram, and it is possible even at very low concentration and samples of your protein as well as the DNA. So the DNA or the RNA is very used in very small amount, because the one thing is that it's very costly to actually have that. So DNA is not costly, but RNA definitely is, and a few nanograms costs about something thousands. So the principle behind INSA is that a purified protein on the cell extract is incubated with your 32P labeled isotope, DNA or RNA, and the complex of the unbound probe is also isolated on the denaturing polypropylene gel electrophoresis, and it's run. So you have the free DNA, you have the protein DNA complex. You cannot see the normal protein because it's not labeled, but if the protein is bound to your DNA, you can see a shift in the band. So after you are running a gel, these bands would separate based on their charge, size, and sometimes shape. So it's a non-denaturing gel, so there is nothing like it's only based on the size and the shape, but here the charge factor also plays an important role. So the free DNA runs fast because it's a very small in size, and then the protein DNA complexes runs, it lags behind, but you can see prominent bands with as they run, like you can see over here. So this is your free DNA and this is also your free DNA, but this has moved a bit, so the intensity of this band is definitely less than this band, and so because it's distributed in between these two bands. Okay, so how do we perform IMSA? So it's the preparation of the cell protein extract, and you prepare radio-labeled DNA or RNA, and you perform the binding reaction. You incubate it for about say 30 to 45 minutes, and then you run it on a denaturing gel electrophoresis, and you study the gel under a autoradiograph or a chemiluminescent or a fluorescent channel. Now, the protein binding to the unique site on the DNA will form a complex PD in equilibrium with the free components. Now, when there is a strong interaction between the proteins and the DNA, that is when Ka is greater than equal to Kd, and you can observe a very distinct band. However, due to dissociation, you would see some a very faint shift. Okay, and the intensity also decreases to some extent, and there is a smear. So smear is basically a drag that you would be observing, and because it's forming a complex and this is an ongoing process, so based on how much smear you are getting, you can tell whether how good the affinity or how strong the binding is. Now, if a single DNA molecule has multiple binding sites for, you say, an individual protein, there will be multiple complexes that forms, and we might observe multiple bands over here. Okay. Now, this is the protocol or the steps that we follow in some. So the first thing is making a fluorescently labeled DNA. So it can be done in your three ways. Yeah, it can be done in three ways. So you have the 5' intact, that is the labeling reaction, and you incubate it with your DNA. Everything is performed in the dark environment and in the presence of ice. So you incubate it, and then it's labeled. So the first one is the 5' OH labeling. The second one is that you are labeling it with a 32p label. So in that way also you are labeling your DNA molecule. So what happens, and it's about biotinylation. You're biotinylating your DNA. Okay. So the second point is your preparation of a gel. So we need to ensure that the gel plate or the spacer on the comms are clean, because this we are studying DNA and RNA. So it's very sensitive, and even a small amount of contamination can ruin your entire experiment. So we should remove any kinds of patches, fingerprints through the glass plate, and it should be cleaned properly with ethanol or methanol, whatever you're using, and should be completely dried before using it. Then we have to prepare the mixture and make the gel, and then you insert the comb immediately, and then you carry out the electrophoresis process. So this is a pre-electrophoresis. So the remove the special comb and bottom, and we mount the gel on a. So it's mounted on a apparatus, the electrophoresis apparatus. We have a gel running apparatus, and this particular apparatus should be kept separate from an SDS gel, because you cannot run an IMSA gel on a SDS or a denaturing gel. So this has to be very clean, very sensitive. So we place the glycerol and the dye solution in each well and conduct the pre-electrophoresis. Now this is basically done to see if the gel is properly formed and it does not break down. Now coming to, after we have pre-run the gel and all, we make the sample preparation. So we prepare the sample and equilibrate for 30 minutes at 20, so it's basically done at 4 degrees or at a lower temperature, and this can be carried out while the gel is undergoing the pre-electrophoresis. Now you load your samples and then you run it on a gel. So you run it for say about an hour or 1.5 hours, and then you study it under the autoradiograph. Now, what do you do? So you visualize the gel bands. So in a lab with dim light. So basically, this entire thing is done in dark condition or with minimal amount of light exposure, such that it does not damage your DNA or RNA. Okay, and you place the film of phosphorus in an exposure cassette, and then you observe it. Now what you see over here is multiple protein binding. So you have the free DNA over. Yes, so you have the free DNA over here, and this is your protein DNA complex that is formed. So you get over here. So the density basically decreases, and what happens over here is that you are seeing another one. So two proteins are basically interacting with your DNA. So this one is a free DNA, this one is your DNA with the protein, and this one is some other kind of protein is also uh, binding to a different sequence on the same DNA. So basically, if you're using short segments of DNA, that is basically very specific. Okay, so if you know the particular sequence of the DNA that binds to your protein, you use only that particular small sequence, say about 6 to 10 nucleotides, and study the and go for IMSA studies. And the other thing is you have the super shift, the super shift assay, where you can see a shift between these two bands. So the shift between these two bands is because some other proteins are coming and binding to our DNA. It's not that only your protein is going to bind to that particular sequence, but there can be numerous number of proteins that have affinity or specificity towards your particular DNA that you are using. Okay. So based on that, you may get a numerous number of bands. So we call that as the super shift, where there is a shift between the original your protein and the DNA complex, and the protein DNA complex along with your protein and the DNA along with some other kinds of proteins that's either because of contamination or maybe because its own nature. So for the control studies, and we can have the protein over here, we can have the specific competitors, then we have the mutant and the non-competitor. There are different labeled probes that we can use, and there are antibodies. So in the first reaction you can have only the probe present. Okay, that's your DNA, so you have over here, and that's fine, so nothing else is present. In the next one you have the probe and the protein, so you can see a particular shift. In the next step you add a specific competitor. Okay, so there also you see that it's not binding with this DNA, but it's binding with a different kind of a DNA, but it's a competitor. So in all these cases you have the probe that is present over here. In the next one you do not have the specific competitor, but you have the mutant. So what happens is that it's a mutant, so it's a non-competitor, so it won't be binding, and you would see a band, two bands over here, and might be you may not be able to see that non-competitor because it's not labeled, and you see a band over here of that you observed for the protein and the probe. And you give another kind of a protein, that's your antibody. So you give the antibody in the last lane, and you see that you are observing a super shift. So in these two cases you see a shift. Now what do you see over here? Let us study the labeled probe. So in this case you have only the probe with a very higher intensity. In the next column, in the next lane, you have your specific competitor, you have the protein and the probe. So your intensity of the free DNA or the free probe gets diminished and you see a band over here, and this is your shift. And same is when you see when you're using a non-competitor, okay, and an antibody. So basically this one you would be seeing. Yeah. And in the middle lane or the third lane you have a competitor along with the probe, so the competitor is not labeled, so you would be only seeing this particular band, because even if it's binding, it's not actually binding. So your competitor is rather binding, so you won't be seeing any band over here, but you would be seeing the same intensity band, or a bit less, you can tell. Might be there is some amount of competition, so might be a very faint band you can see over here, and that's a sign of competition. And when you're using the antibody, there is a shift. You might not observe one particular band, there might be another band as well, but a very lighter band, because mostly your both the antibody and the protein would be binding. So in the higher intensity band would be this, you would be seeing some free probe of the DNA as well, and a faint band like what you see in the competitor you might observe. So in this way, you can frame your control studies and understand different ways of explaining your INSA studies, or how your protein is binding, or has better binding or worse binding to your DNA. So for that you can also do some comparison studies, and you can calculate the dissociation constant, association constant, and you can comment on the strength of you can comment on the binding affinity of your protein and the DNA or an RNA, whatever you take. So how and why this titration is done? So as we know that we are going to have a DNA or an RNA that's interacting with a protein, and for that we need to have enough DNA and protein in the solution, but we know that it's a very sensitive process. So the amount of DNA or the RNA that would be required is less. So this is the titration profile of a 16 residue single-stranded DNA with a human AGT protein. So you see that these lanes have a decreasing DNA content, and here you have the increasing protein. Okay, so you see the free DNA, free DNA, free DNA, and you are seeing that the complex is starting to form. Okay, so this is an increasing DNA complex, and this is a decreasing increasing protein. So with the increasing protein and the DNA, you can see that the band intensity gets thicker and thicker. And this is your protein DNA complex, and the A is your this F is your free DNA, and this is the bound complex. So these ENSA have different applications in understanding the conformational changes that you see in the DNA, and maybe also for from the perspective of a protein. For example, if we are running an IMSA for a 2D variant with the electron microscopy, you can understand whether the DNA is changing its conformation or the protein is changing its conformation, and why the binding is getting weaker or stronger. So it helps in studying the conformation or characterization of the conformation. Also, it helps you to analyze different cellular extracts for the presence of certain DNA binding proteins or different transcription factors, and you can catch up with them to undergo to help to study the regulation of different transcriptional machineries. And you also so the third point is that it proves useful for different stoichiometric analysis, so you can get an idea of the number of proteins that is binding to that particular DNA sequence, or vice versa. Okay, so it might happen that a number of DNA sequences that binds to your protein. So in either way you can study your protein nucleic acid interaction. So it might not, it's not only with the DNA, it can be with the RNA, but the protocol or the principle remains the same, only your DNA changes to RNA. Now it is also used to study whether your transcription factor is attached to a DNA or not, and if the transcription factor is attached or not, you can study their activity, the binding affinity and all. So this particular technique has numerous advantages. A few of them includes that it can be used in wide range of nucleic acids of different sizes, structure, as well as you can use a wide range of proteins. It's a relatively simple method to perform and can be used for your qualitative analysis. So you can study the different bands via different softwares you have, and you study the intensity, and you can understand at what concentration your particular DNA is binding with your protein, and you can study the KD from there. That's the binding affinity. So it's a very nice way to measure the you can to analyze it qualitatively. Okay. Now also it proves very useful in the detection of protein that binds to your DNA and cause difficulty in migration of fragment on the gel. It's a highly sensitive method, and usually it's of low cost, so you do not require any special equipment. And in this technique it's possible to use both the crude extract and purified recombinant proteins. So also this technique faces a few drawbacks. So it does not provide a straightforward measure of the weights of the proteins, as mobility is influenced by several other methods. So you do not get any idea about the molecular weight of the protein, or what proteins are actually coming and binding to your DNA sequence. So it's not an appropriate method for the kinetic studies. The dissociation is also one of the major drawback of IMSA. It occurs during electrophoresis, thus preventing detection. So you have to be very careful with all of this, and it's a very sensitive method. So any kind of contamination can affect your DNA or RNA. If it's RNA, it's very difficult to study different to perform different studies. And interacting biomolecules must have different electrophoretic mobilities. So if you're studying the same kind of biomolecule, it becomes very challenging. Okay. So in conclusion, so we can tell that it's used for purposes such as quantifying interactions between DNA, RNA, and your proteins. It works on the observation that protein-bounded DNA migrates slowly as compared to your free DNA, and your electrophoretic mobility shift assay is most widely used method for the detection of protein DNA interactions. So that's all about from the theory portion on protein carbohydrate interactions and protein nucleic acid. But this is only the theory part. When I will be so I will be starting with the questions, and then we can talk about a few more other concepts. So there are questions about the different other kinds of glycoproteins, glycolipids, and a lot more about protein nucleic acid interaction. So we can do that. So okay, so let us take a 5 minute break, and then we can discuss with the questions.
Sure, sure.
Okay, so let us solve the questions. Let's see. Okay, so this is the first question. Let's see. So first you answer, and then I will be telling the right one. So it is saying which among the following is or are true about proteoglycans. So they are present in both extracellular and intracellular matrices. Only one sulfated glycosaminoglycan chain is linked to the membrane protein. This glycosaminoglycan is connected to the serine residue through a tetrasaccharide bridge, and the heparan sulfate and chondroitin sulfate are types of proteoglycans. So, which of the following do you think are correct? Is or are correct? You can write in the chat box. You say C. Okay, so let us see. They are present in both the extracellular and intracellular matrix. This one is correct. Heparan sulfate and chondroitin sulfate are types of proteoglycans. This is also correct. So 1 and 4 are correct. And if you see only one sulfated glycosyl aminoglycan chain is covalently connected to the membrane protein, this is wrong because only one, no, it can be more than one. It can be one, but not only one. It can be more than one. So this is the incorrect option. And 3 is also correct. It is connected to the serine residue through a tetrasaccharide bridge. So 1, 3 and 4 are correct options. So C is correct.
Technically, heparan sulfate and chondroitin sulfate are GAGs, right?
Yes.
So GAG is a part of a proteoglycan, but can you call it a proteoglycan itself? I was confused about that. I guessed it, but
Just because there is no option 1 and 3, so I believe you can throw that in that
yeah,
particular
maybe I
case.
thought there was no better option, but otherwise I would just consider it a part of a proteoglycan.
Yes, it's actually part of that. But it's okay. It's part of it means it's a type, falls under some category of it. The next one. Oligosaccharide microarrays are used to determine. So, what do you use oligosaccharide microarrays for? Specificity and affinity of carbohydrate binding to lectins, amount of carbohydrate bound to lipid membrane. This is not correct. Okay, affinity of a particular amino acid residue to proteoglycans, the amount of glycation in a protein. Yes, so it's used to identify proteins with specific affinity to particular oligosaccharides, like you have the carbohydrates which is binding to the lectins. Yes, so A is the correct option. Very good. The next one, uh, glycosphingolipids. Glycosphingolipids are the plasma membrane components in which the dash head group and R dash. So its hydrophilic or hydrophobic head groups are monosaccharides, oligosaccharides. Which one? So glycosphingolipids. You have a sphingosine over there. You have the glycol, and you have the fatty acid chain, and you have the sphingosine, and that's bound to some portion. So the head group Yes. So the head group is basically hydrophilic and B, so oligosaccharides. Yes, that is the correct option. So if the head group is hydrophilic and these are oligosaccharides. Correct. Okay, the next one. Which among the following structural motifs is generally not found in RNA binding proteins? So which of the following motifs will you not find in RNA binding protein? So you have the RRM, the RNA recognition motif, you have the double-stranded RNA binding motif, you have the zinc finger motif and the helix turn helix motif. So one of the above is your DNA binding motif. So if you can find the DNA binding motif, then you can tell the right answer. So which is not a RNA binding protein. Okay. Yes, so the RNA recognition motif is correct because it's recognizes the RNA. It binds to RNA. Then you have the double-stranded RNA binding motif that is also RNA binding protein. Now the zinc finger motif is mostly important for your RNA packaging, then different transcriptional activation, regulation of apoptosis, then you have protein folding, lipid binding, and different assemblies. But your helix turn helix motif is basically a DNA binding motif. So D is not a RNA binding protein, and this is a DNA binding protein. So helix turn helix is the correct answer. Good. Now the next one. HbA1c test is a method to detect the modified form of hemoglobin. Which among the following statement is or are false about this and or the modified form of hemoglobin? It is used to measure the average plasma glucose concentration over prolonged periods. It is a form of hemoglobin that is chemically linked to sugar and it is formed through an enzymatic pathway. Let me see. Okay, you gave the answer D. Okay, so it is formed through an enzymatic pathway. So this HbA1c test is basically used to measure the amount of blood sugar or the glucose that is attached to the hemoglobin. So this modified form of hemoglobin is basically called as the glycolated hemoglobin, which is formed through non-enzymatic glycation pathway, which is caused by the interaction of the sugar, that is you have the glucose with your hemoglobin. So it's formed by a non-enzymatic glycation pathway. So basically number 3 is the incorrect answer. Means
So
the
how does glycation vary from glycosylation? I assume there's a glycosidic linkage in glycosylation.
glycosylation
Yeah.
and so glycosylation is you can tell it a kind of a reaction in which you have a carbohydrate, okay, that is
Mm.
the glycosyl donor that is attached to a hydroxyl or another kind of you can say any other functional group. It can be an NH2 say of a molecule in order to form a glycoconjugate. So it basically, in if you speak of glycosylation, it is an enzyme catalyzed reaction, whereas glycation is a non-enzymatic reaction.
So the enzymatic activity is the only thing different, right? Biochemically, it's the same. Otherwise,
Otherwise, it's basically the same. The
right.
enzyme plays a role over here. So D is the correct answer. Okay, the next one. This is an easy one. Dash type of lectins are essential for cell-cell adhesion. Y type lectins for development of maintenance of nervous system, and Z type of lectins are important for their roles in generation functional lysosomes. So I, P, and C. So which one will be which? So the I type, P type, and the C type. So cell-cell adhesion will be easy to solve. Maybe the next two becomes tricky. Okay, you have an A type as well. Okay, A type. You have a C type, A type, P type, and I type. So for cell-cell adhesion, because you have the calcium that's playing an important role over here. Yes. So because of that, the calcium, you have the cadherines that becomes activated in the presence of calcium. So X is your C type. Okay, then you have then the Z already becomes your I type, where you have the lectins that are uh, that plays different roles in the generation of your lysosomes. But A or P, so you would see that Y type of lectins, that is the P type of lectins, are basically important for the development and the maintenance of your nervous system. So what is the role of A type of lectins? So So basically, it has also different roles. I'm not being able to recollect what it has, but anyway. So P type of lectins are mostly for this nervous system maintenance and all. Okay, the next one, which of the following is not true about the RNA's footprinting assay? So proteins that bind to the RNA sequence can protect. So, which of the following is not true about RNA footprinting assays? So, proteins that bind to an RNA sequence can protect the region of the RNA from RNA's digestion. The region that is not protected is known as the footprint of the protein. This makes it possible to locate a protein binding site on a particular RNA molecule. And you have this method uses RNAs to cut the end labeled RNAs with and without bound proteins, followed by gel electrophoresis to detect the cleavage pattern. Yes, because everything else was correct, because the proteins that bind to the RNA sequence can protect it, and in a similar pattern, if you say that the region that is not protected is the footprint, that is incorrect. The region that is protected is basically known as the footprint of the protein. So this sorry, which is not true, right? So basically B is the incorrect answer. Yes, good. The next one, interaction of proteins with nucleic acids is essential for which of the following biological processes? So it's a question from the functions of protein nucleic acid interaction. DNA replication and repair, RNA processing and translation, gene expression, and regulation of chromatin structure. So what is which one? So RNA processing and translation, this is correct. Gene expression, that is also correct. Regulation of chromatin structure. Okay, so the interaction with the proteins, yes, that is also correct. You can regulate it because you have the histones, okay, and DNA replication and repair. This is also correct. So all of the four options are correct. So in which we have 1, 3, 1, 2, 3, 4, and 1, 2, 3, 4. Yes. So D is the correct option. Very good. Okay. So all of the things are important for protein nucleic acid interactions. Okay. So which among the following is not a function of replication protein A? It prevents single-stranded DNA from winding back on itself or from forming a secondary structure. It helps to protect the single-stranded DNA from being attacked by endonucleases. It also binds to DNA during the nucleotide excision repair process, and it regulates the transcription of genetic information from DNA to a messenger RNA. So basically, this replication protein A is okay. Let me see what you people are answering. Then we can study. So it has different subunits. Okay, so it has a 70 kilodalton subunit. It has a 32 kilodalton subunit. It has another subunit of 14 kilodaltons, along with it has different folds. Basically, it has 6 folds. Oligonucleotide, oligosaccharide that enables the binding of oligonucleotides and oligosaccharides with different DNA binding domains. So this replication protein A is a major protein that basically binds to single-stranded DNA in your eukaryotic cells. In different in vitro studies, it has been shown that it has higher affinity for single-stranded DNA than RNA or a double-stranded DNA, and this RPA is very required for your replication and the different recombination processes, different repair machineries, and like you have the nucleotide excision repair and the homologous recombination, and it also plays roles in repairing of the damaged DNA. So it prevents what are the options? It prevents the single-stranded DNA from winding back on itself. That is correct. It helps to protect the single strand from being attacked by the nucleases. Yes. It also um, binds to the DNA during the nucleotide excision repair process. Okay. And it regulates the transcription of genetic information from DNA to messenger RNA. So yes, which is not a function. So yes, D is not a function of a RPA. Yes, because these transcription factors are the proteins that's regulating the transcription of genetic information from DNA to messenger RNA, and it's not a function of your replication protein A, right? So it has to be done by a transcription factor. Okay. The next one. Syndecans and glypicans are two major families of membrane heparan sulfate proteoglycans. So dash are held by, so syndicants or glypicans are held by hydrophobic or hydrophilic interactions. between a sequence of polar or non-polar amino acids and plasma membrane lipids. However, glypicans or syndicans are held by covalently attached membrane lipids. So two important um, part of the family of heparan sulfate proteoglycans. So basically your syndicants have a transmembrane domain in their core proteins, and your glypicans are attached to the cell membranes by glycosyl phosphatidylinositol anchors. And your glypicans have roles in the development of modulating morphogen gradient formation and cell growth by regulating different signaling pathways, and these syndicants function as your co-receptors with the growth factors or your adhesion receptors, and it tunes different signals that is transferred across the different cells to the cytoskeleton or the cytoplasm mediators, and affect the activation of different signaling cascades. So what are you answering? You're answering it as A. Okay, so syndicants are held by hydrophobic interactions, that is correct, and then between a sequence of non-polar amino acids, right? And however, glypicans are attached to the membrane lipids. Yes, that is correct. So A is correct answer. Okay, the next one. Histones have a dash content of dash and dash as well as dash charged amino acids. Wow. So histones have and dash as well as dash choose amino acids. Okay, let us see. So low or high aspartic acid or lysine or valine, and glutamic acid, arginine or histidine, and positively or negatively charged. So histones are basically positively should be positively charged. Yes, so I feel that is correct. And then you have a lysine and arginine, so they have a high quantity of positively charged amino acid, right? So they have a high amount content of positive. So lysine arginine low, not, so B is the correct answer. Yes, high content of lysine and arginine are any positively charged amino acids. Yes, that is correct. Okay, the next one. Which of the following methods are used for the detection of protein nucleic acid interactions? So we have spoken of one type. So what can be the other types? You have the pull down assay, you have the IMSA, you have tRNA footprinting assay, and the chromatin immunoprecipitation. So there can be other techniques as well. So what can you use? Protein nucleic acid. So 2 is correct. So in all of them we have 2. Okay. We have 3, 1. So in this pull down assays, what you do is you isolate a protein complex by absorbing with a particular complex onto beads. So these beads are to these beads you have different ligands that are immobilized, and that helps to catch your protein, and then you can elute them. Okay, so that is basically an in vitro technique for detecting physical interactions between different molecules. And this tRNA footprinting assay is for when you study for tRNAs. What about the chromatin immunoprecipitation? So chromatin immunoprecipitation, it's an antibody based technology used to selectively enrich specific DNA binding proteins along with the DNA targets. But what about the tRNA footprinting assay? Will it be, can it be used to detect protein nucleic acid thing? So basically, if I can recollect and tell, so for the tRNA, basically it's a protein nucleic acid interaction. So yes, so tRNA footprinting assay, so it's a nucleic acid. It's not specifically telling about a DNA or RNA. So yes, you can use this tRNA footprinting assay as well to detect your protein nucleic acid interactions. So yes, 1, 2, 3, and 4, all of them would be the correct answer. And the last question for today. A DNA binding tetrameric protein with four identical subunits operates by a helix turn helix motif in its DNA binding domain. Good. Which inhibits the expression of genes coding for proteins involved in the metabolism of disaccharide in bacteria by reducing the transcription of proteins. What is the name of the DNA binding protein and how does the bound repressor reduce the transcription of a protein? It's a lambda repressor protein, it's a lac repressor protein, it's a lambda repressor or a lac repressor, and functions by prompting DNA looping only, by only obstructing the RNA polymerase binding, and DNA looping obstructing the RNA polymerase binding. Okay. Okay, so what are you telling? It's a lac repressor protein and it functions. Yes, that is correct because a lac repressor is a DNA binding tetrameric protein that has four identical subunits and it also operates in a helix turn helix motif in its DNA binding domain, and it inhibits the expression of genes that codes for the proteins in the metabolism of lactose in bacteria, and this repressor that is bound, it reduces the transcription of the lac proteins by occluding the RNA polymerase binding site or by the prompting of the DNA looping. So yes, D is the correct option. Good. So that's all with the question and answer session. So I think if you have any questions, you can ask, or else we can end the class.
Yes, I wanted to clarify about the one concept of uh, proteoglycan, glycoprotein, and lectin, like with a simple like point-wise annotation. जितना मेरे को understand हुआ, lectin is a specific protein with CRD or carbohydrate recognition site,
Yes, that
and
is
it's
right.
referred to the protein alone.
Sorry.
Uh, yeah, lectin is a specific protein that binds to carbohydrate.
Yes.
So even without the carbohydrate, it will be still be classified as a lectin.
Yeah.
Its job is to bind, as in a lectin or anything like that.
Yeah. Yeah.
Then proteoglycan is bound with the GAG,
Right. Yes.
and glycoproteins are bound by smaller oligosaccharides, and it's a bit more like globular or like clustered
You're telling
compared
of
to
glycoproteins.
yeah, the major difference between proteoglycan and glycoprotein.
So these glycoproteins are basically like you have the N-linked and the O-linked.
Right. The method
Yeah.
is different.
So they are bound to the, you can say the terminal ends. and they are also linked to the carbohydrate chains.
All right. So, okay. So the proteoglycans necessarily need to be connected via tetrasaccharide linker.
Right.
So I can make a point of GAG tetrasaccharide linker, and yeah, that's the major difference, right?
Yes. So
And you won't call the rest of the protein part as a lectin. Like for example, if I have a glycoprotein, since the protein part is binding to the specific oligosaccharide, won't that make the protein part the lectin for the glycoconjugate? The
it's very difficult to say because a protein that is binding to carbohydrate, you call that a lectin.
yes,
Yeah. So if it is binding, say, what it binds to a sugar.
right,
So very difficult to actually classify. Okay, from the definitions, you put them in different categories. That's also fine.
all right. Just a matter of like general categorization.
Yeah, but it's very difficult to actually tell from the definition what it actually tells about it, because any protein that binds to a carbohydrate is actually lectin. So why there are proteoglycans and there are glycoproteins? Yeah, that's a valid question.
All right.
Okay then, that's all then. Thank you. We will meet in the next week.